every week. So we've known John, I've known John for many, many years. And in fact, John did a research assistantship at the Brigham and Women's Hospital and Harvard Medical School just before I went there. So he knows what it's like in the labs where I did my training. And I feel like you just basically have some kinship with anyone who's been through that rigmarole of getting, shaving and getting prepared to walk into a lab in dark light or dim light at about 3 in the morning and pretend that it's not 3 in the morning for your participants who are staying in a lab a dim light environment for about a month. So that kind of brings some kinship, and I was delighted when I came to Oregon that John was here, and he's been helping with all of our research projects, and actually he's a PI, a multiple PI on one of the RO1s that we started himself. John has had excellent training with both at the Brigham and Women's Hospital and also with Al Louie, as Al was on the call just a few minutes ago, if you all heard him. John had a K-23 grant from NIH on genetics of morning and evening types in the blind and sighted people. His first two papers, the first one was in the New England Journal of Medicine. The second one was in the journal Science. And, John that's a tough act to follow, but John has been publishing at pace since then. And apart from all of those wonderful experiences that I've had in the lab, I just want to tell you that every time we meet in our lab meetings, you'll learn something new about John. But one highlight that really stood out for me, apart from the sort of very strange video sketches that sometimes he'd show at lab meetings, this was before TikTok existed. But I was most impressed when John was able to draw a map of the 50 states of the USA in less than 5 minutes, with only a little bit of help from Matt Butler. Of course, I'm from England, Great Britain, and I'm not quite sure if I could draw Scotland, but John was able to do all of the, the panhandles with the right ratios and everything else. It was just so impressive. So apart from his scientific prowess, his good humor, his great camaraderie, he's a good man in a crisis, and we love him. And John, over to you.
Thank you very much, Steve. That was very, very, very kind of you. And I think the one thing I would say is I remember Chuck Allen and I, when we realized you would be coming to be the director of the Institute, we were sitting in Chuck Allen's car together, and we both had this kind of excited moment like this.
You are in the...
So the feeling is quite mutual. So let me share my screen, and let's get in presenter mode here. All right, so I'm going to be talking about a disorder called non-24-hour sleep-wake rhythm disorder. And I stole my title for myself. I kind of plagiarized, and this was an editorial I wrote for actually a paper of Andrew McHill's, which is kind of relevant, and I'll actually be showing a little bit of Andrew's data since I love his camping studies so much. But I want to talk about just a quick intro to circadian pacemaker for those who kind of aren't aware. And then I want to talk about this disorder that shows up in both blind and in sighted people. I'm mainly going to be talking about this disorder in sighted individuals. And I'm going to be talking about a paper that Al and I just published along with colleagues back in Boston on non-24 in sighted individuals. And it's really just a series of 3 case studies, but I think it's still very illustrative. So that will be, I suppose, on some level, the less rigorous part of the talk. And then I just want to finish off a little bit talking about what I am proposing as a revision to the nosology, the classification of these disorders, which I think is going to be important. So just really quickly, when we're talking about these rhythms, these endogenous rhythms spontaneously generated by the body with a periodicity of about 24 hours, That is going to be really important when we talk about these non-24-hour sleep-wake rhythm disorders, because we don't have a 24-hour day. And so this is the more recent collection of data from the Boston group showing an average period of just a little over 24 hours. Differences between males and females, but a nice normal distribution of the length of our biological day. And of course, you can measure this in a whole bunch of different parameters. But most importantly is that these rhythms are all at least internally synchronized by a clock in the hypothalamus, which is at the base of the third ventricle, you can see there. And importantly for our discussion today is that these rhythms are synchronized or entrained to the 24-hour day by stimuli in the environment, most importantly, the light-dark cycle. So this is a big review for many people. But I think it's important to kind of talk about what do I mean when I say synchronized or entrainment, and how would I measure that? And we're going to get to that, but just as a reminder, the way this is happening is through intrinsically photosensitive retinal ganglion cells, as well as rods and cones that are transmitting that light information from the eye to our 24-hour body clock. All right, so let's talk about entrainment a little bit. So I'm just showing here, this is the onset of melatonin secretion. So this was, of course, developed by Al Lewy, invented by Al Lewy, as a measure of biological timing. So you can see the clock hour, and we're not catching the full 24-hour profile of melatonin secretion. We're just getting that onset gathered in dim light to avoid the suppressant effect of light. And that little red dot is just marking when it crosses a threshold, in this case about 3 picograms per mil, gathered in saliva hourly from this participant. So this is a healthy, normal person, and this is day 1. And this person came back 2 weeks later, and their melatonin onset was about the same time. They came back another 2 weeks later, it wasn't that different. Another 2 weeks, and it had moved maybe slightly earlier. So you can think of this onset of melatonin secretion, or at least I like to, as marking the beginning of the biological night. So it's stable, it's not moving a whole lot. It's on a 24-hour rhythm. It is therefore entrained to the 24-hour day. And we can replot that same data, only this time instead of the y-axis of plotting the melatonin concentration, now I'm just plotting day of study, moving forward in time as you go down. And so this is the exact same data I just showed you, only replotted. So you can kind of see that I'm getting a vertical line. The timing of the clock is staying at the same clock hour. And that's what the MO stands for. This is the dim light melatonin onset. So it's stable. That's entrainment. And most of us, presumably, are entrained. Just like most organisms on this planet, we're all entrained to the 24-hour day, and for most of us, it's that light-dark cycle that's doing it. But if you take any of us, anyone who's listening right now, and you put us in an environment free of time cues, where we're not being exposed to the 24-hour regular time cues, we don't stay entrained. So this is an example of a person we studied back in Boston. And what you see here is, again, a clock hour on the x-axis, so it's 24 hours of time. We're moving forward in time. Each tick mark is a day as you go down. And the black bars represent the timing of sleep. And you can see this is a male, in this case, his sleep-wake timing drifts to a progressively later time each day. The dashed line is actually marking his the minimum in core body temperature, which again is also moving later every day. And we typically will plot this data because it's crossing the midnight line. We'll double plot it, meaning we'll plot successive days, not just beneath each other, but next to each other. So you can really get a sense of what's happening as he kind of crosses midnight in terms of his sleep timing. And hopefully you can see my cursor, but if you look in the middle of this section, you'll also notice something interesting happens to his sleep timing here, there's this general rate of drift, but all of a sudden you can see he makes these big leaps where his sleep-wake timing might drift 8 hours later every day before he settles into this kind of slower rate of drift of maybe about 3 hours later every day on average. And you can see this if you have someone in time isolation, again, and living in a laboratory where they are unaware of time and aren't getting any external time cues for even longer periods of time. So this is an older study from Chuck Seisler. And again, what you can do is you can double plot or triple or quadruple plot the same data. In this case, it's triple plotted, and you can really get a sense of how that sleep-wake timing is drifting progressively later every day. The kind of tan stippling, again, this is sleep-wake timing in dark, but the lighter colored stippling is when the core body temperature is below average. And again, you'll see times where the sleep-wake timing really, going back here, really drifts much faster. Again, I'm looking around day 50. You can see sleep-wake timing is drifting 8, almost 12 hours later a day. So it's moving very, very quickly and then assumes a slower rate of drift. And you can see when it does this, it breaks away from the temperature rhythm. So that's pretty weird. And we're going to talk about that more. All right. So that's a little bit about what happens to all of us when we're entrained and what happens when you take away those time cues, or you just let us be in charge of the time cues, because in this sort of environment, the participant was deciding when they were going to turn their own lights on and off. So let's talk about non-24 in blind individuals. So in the blind, and when I'm talking about the blind, I'm talking about people totally blind, so they have no conscious light perception. The majority of those folks are going to have circadian rhythms that are not entrained to the 24-hour day, and I'll show you what that looks like. And as a result, all of the rhythms under the control of the clock, including sleep-wake propensity, will drift to progressively later time every day. And as a result, they're going to have these periodic symptoms of insomnia and daytime somnolence when their clock is out of sync that will kind of relapse and remit when they get into sync. And again, I'll show you what that will look like. So this is 24 hours, again, in this case, this salivary sampling for melatonin. And this is a totally blind individual. She actually doesn't have eyes. She was bilaterally nucleated, meaning her eyes were removed. And so Al and I studied her a long time ago. And you can see she's making her melatonin at night. So this is plotted in an odd fashion, and I'll explain, it'll become clear why. But you can see she's basically making melatonin at a normal time. And again, that red dot is marking the onset of her melatonin secretion. So everything looks fine at this point. But what you're going to see is that the timing of her clock, as marked by that red dot, that onset of melatonin secretion, is going to drift progressively later every day. So here we are 2 weeks later. She's drifted a little bit later still, slowly drifting through the night, again drifting even later. And now you'll look, she's making melatonin. Her melatonin onset is at 9 in the morning, roughly. So when she goes to work at her job, and she worked full time, she's going to have trouble staying awake because her body thinks it's nighttime all throughout the working day. And conversely, when she gets home at night, even though she's been awake all day, she's going to have trouble sleeping because her body clock is trying to wake her up. It thinks it's daytime. So she's going to be most symptomatic here. But it doesn't stop. It just keeps going. She's moving a little bit quicker here across the day. Before 83 days later, she ends up exactly where she started. Now her symptoms will probably remit. Now nighttime is the biological nighttime, corresponds to when she wants to sleep, and she's going to be able to sleep well and be awake during the daytime. And similarly to the way I replotted it before, we can replot this again instead of plotting the melatonin concentration, we can plot with those red dots that onset of melatonin secretion, that beginning of the biological night, moving forward in time on the as you go down the y-axis. And I'm just going to replot that same data as I did before. And you can see day after day that rate of drift. She drifts progressively later every day. It's not a constant rate of drift, but on average, she's drifting about 16 minutes later every day. And coming up on a decade ago, we published, I think, what would be the most comprehensive assessment of non-24 individuals, blind individuals. And you can see both a lot of inter and intra subject variability in the rate of drift and the constancy of that rate of drift. But these are all melatonin onsets for separate individuals. And you can see most of them are drifting progressively later. Those in red are actually female, and those in gray or black are males. And again, like I was saying before, most totally blind individuals, those who have no light perception, have this non-24. So this is a individual I studied very many years ago when I was working as a research assistant. And you can see, again, the black bars in this top graph are sleep timing, and the X's represent the, with a circle around them, represent the endogenous minimum and core body temperature. And you can see the clock drifting progressively later every day. And what we did is we exposed this individual to light, trying to see if light information could be transmitted from the eye through to that hypothalamic circadian pacemaker, and then onward through a very circuitous pathway all the way to the pineal gland, where it would exert its suppressant effect on melatonin. And that didn't happen in this case. That light information wasn't getting downstream to the pineal gland, and they were not synchronized. And so this is a case for most totally blind individuals, those with no light perception, but not all. So this individual, they also have sleep that's pretty constantly timed, but you can see their melatonin onsets in these open triangles are occurring at the same time from day to day. And when we expose them to bright light, which, by the way, Al Louie was the first to kind of show the suppressant effect of light on melatonin, they have this robust and very normal response to light. So that photic information, despite the fact that they had negative electroretinographic responses, no visual evoked potentials in response to light, and we confirmed it was going through their eyes, and obviously no actual conscious light perception. Despite all of that, they had enough intact photic input to keep their clock synchronized or entrained. Again, we've got these melatonin onsets occurring vertically here. But although most people who are totally blind have non-24, most individuals with non-24 who are blind don't have sleep and wake timing that drifts progressively later. It may happen, as that first bullet says, but most actually maintain a 24-hour of schedule of sleep and wakefulness. Now, if I go back, you'll notice this individual is keeping a nice 24-hour schedule. They're entrained. But if we go back to this previous individual, despite the fact that he's not entrained, he's maintaining a very regular, spontaneous 24-hour schedule of sleep and wakefulness. So that's what most blind individuals are going to do. But why do we think that it's a non-24-hour sleep-wake schedule disorder? I think part of this goes back to one of the earlier papers. So this is a blind individual. And in section A at the top here, he was, I think, living at home. And you can see the gray areas just when he's sleeping. And then they brought him in section B here. and let him, he was not in isolation, he knew what time it was, but he could do whatever he wanted. He had this ad lib schedule of sleeping and waking. And you can see he has this very nice drift to a later time in his, this again is his sleep. But then in section C, when he returned home, he readopted a 24-hour schedule of sleep. You can notice that his napping is drifting a little bit later across the day. So you can see that it's probably likely that his clock is not synchronized to the 24-hour day. He's kind of trying to force himself to try and sleep on a 24-hour schedule, and his sleep is obviously disrupted. But that's what he's trying to do. And if I was just looking at section C in isolation as sleep diary, it might not be obvious to me that he had a non-24-hour schedule of sleep and wakefulness. It wouldn't be as obvious as what we see in section B, when he was allowed to do whatever he wanted. And in fact, this is again what most blind individuals with non-24 do. They keep a very much on a 24-hour schedule. So this is a couple of years of data. This is wrist actigraphy. So this is a glorified Fitbit. We're measuring movement. So in this case, the dark area doesn't represent sleep, it represents movement, and the lighter colored is not moving. And you can see, you can actually, if you look closely, you can see weekends, because if you'll see, there are these, every 7 days, there's like a little gap where she sleeps in. She doesn't have to get up so early to go to her job. She had an early job that she had to start. But this is a blind individual who is not entrained. Her body clock is drifting later, and yet she's maintaining this 24-hour schedule of rest and activity. But you might also notice some activity drifting across the day. And we can detect that if we do a spectral analysis of this data. And so when you do that, if you chop up this entire 2 years of data into period lengths between, say, 23 to 25 hours, and you're looking for kind of where do you see the least variability, you can kind of see she's mainly on a 24-hour schedule, although she does have another peak at about 24.15 hours, which corresponds very nicely with what her clock is doing, what that pacemaker, that drift. And I can show you another example of that that's a little clearer. Here's another individual. Again, the dark is when he's active, the light is when he's trying to sleep. And the triangles represent that drift to a later time in his melatonin onset. So he's clearly not entrained, it's drifting later every day. But he's trying and basically maintaining a 24-hour schedule of rest and activity, and probably sleep and wake. And again, when we do the spectral analysis, we see mainly a 24-hour schedule, but we do see his rest activity also has this 24.3-hour component to it, which corresponds perfectly to what his underlying pacemaker is doing. It's drifting about 0.3 hours later every day. So now I'm going to shift gears and I'm going to talk about the sighted. So this is one of the first papers looking at sighted individuals, a sighted individual whose sleep drifts progressively later every day. And this was described as a hypernycthemeral sleep-wake schedule. And you can see, again, it's double plotted. And you can see it's many months of data. And you can see, although it's not at a consistent rate, his sleep-wake timing is drifting progressively later every day. Except maybe near at the end here where he has, maybe a week or so of more of a 24-hour schedule. And it's not just his sleep that was drifting later. When they looked at his core body temperature, so this is actual plots of body temperature, you can see that minimum in body temperatures slowly drifting later as well. Some of that's the masking effect of his changes in posture and sleep itself. But this was unusual, and it was thought when you look at that, and here's just, there have been many case examples of this. So this is again sleep diary data from 2 individuals, sighted, whose sleep is drifting progressively later every day. And people said, hey, we've seen this before. We know where we've seen this. We've seen this in the blind people. Again, if you look at the two individuals on the right at their sleep-wake timing, and you look at section B on this blind person, they look really similar. So they called it eventually the same disorder. They all have non-24 hour sleep-wake schedule disorder, sleep-wake rhythm disorder. But remember, this was an ad-lib schedule where he could do whatever he wanted. And what's probably more reflective of the way the blind present is, like I said, that where they're trying to maintain this 24-hour schedule, and primarily do in terms of their sleep and wakefulness, even though their clock might be drifting. So there are already some clues that these look a little bit different, even though we've given them the same name, and superficially on some level, they seem to be the same disorder. The other thing that's odd about this in the cited is the rhythm, the periodicity of their sleep-wake schedule. So this is the distribution of that period of their sleep-wake timing. And you can see it's obviously not normally distributed. It's got that nice skew to it. But most of them have periods well above 24.5 hours. This is from a Japanese series of 57 of these individuals, the largest in cited people to date. That looks a little different, right? Most of these folks are above 24.5. That's not the same as the period of the human circadian pacemaker, either in its distribution or its average. So again, most of these folks, their sleep is drifting at a rate half an hour later every day, an hour, 2, 3, 4, 6 hours later every day. And as I showed you, sometimes they might even drift 12 hours later a day. That's not what our endogenous circadian pacemaker does. It's ticking away at a much slower rate. So what could be causing this in the cited? Well, it may be that their period is just too long. You know, maybe the reason for this long periodicity of their sleep-wake rhythm is because, well, they're way out of this normal distribution. They've got a period so long that they can't hook on to the 24-hour day. They can't entrain to that. The other possibility is maybe they're kind of the mirror image of some of those blind individuals. They can see just fine, but somehow they've managed to lose input to their 24-hour body clock, that light input to their clock. Now, physiologically, that's kind of hard to figure out how that would happen. Those intrinsically sensitive retinal ganglion cells, well, the retinal ganglion cells, the rods and cones project to that. So you would have to figure out some very odd way to knock out input to your occipital cortex so you can't see, but somehow preserve that circadian photoreception. But it's an intriguing theory nonetheless. It may be that they're differentially sensitive to the light, though. Maybe they've got less sensitive to the morning light, which will phase advance or shift us to an earlier time. So they're not getting that corrective shift in our clock that we all get every morning when we turn on the lights or go outside. Or maybe they're more sensitive to the light that we get in the evening that shifts our clock to a later time, this phase delaying light. It may be that there's actually a disassociation between what their sleep is doing and what their body clock is doing. They may have broken apart. It may also be that it has nothing to do with our 24-hour body clock. Maybe it has to do with how we build up sleep drive. That's called homeostatic sleep drive. So you can think of this as the longer I'm awake, the more sleepy I get. Well, maybe these people with non-24 who are cited, they take forever to build up enough sleep drive. I can build up sleep drive just 16 hours of being awake, but maybe it takes them 20. And so we get this huge buildup, a huge amount of time it takes us to build up enough sleep drive. And then maybe also, and not mutually exclusively, it takes them forever to pay off that sleepiness. So after 8 hours of sleep, I've used up all that sleepiness. Maybe it takes them 12. So everything is going to shift later every day. And then the last piece is that maybe it has to do with their self-selected pattern of light exposure. Maybe that's going to affect the clock in the absence of any frank circadian dysfunction. And I'm going to argue heavily for the fourth and sixth bullet points. And that's what we did in this paper that just came out in the Journal of Clinical Sleep Medicine. And we studied just a small number of individuals. These are just a series of case studies. So I can imagine folks kind of rolling their eyes about making dramatic conclusions based on 3 people. And I think what we were doing is really trying to say that these are folks who would prove the point. So the first individual we studied was a 31-year-old man who had kind of spontaneously adopted a 24-5-hour day length when he was in his early 20s. And so the first part of the study was just to have him keep a sleep diary for us and wear an app to watch, something to measure his actigraphy. And we saw, that's what's circled in red here, is that he had a sleep-wake schedule that had about a periodicity of about 25.2 hours. So he's drifting about 1.2 hours later every day on average. The green is a simulation of what time it is in his body, a kind of simulation of the melatonin onset. Then we brought him into the lab, and we did a very complicated protocol, that's what's in the lighter gray, where we assessed what was the length of his biological day. And it wasn't 25.2 hours, it was 24.5, certainly well within the normal range, on the longer end of the normal range, but certainly compatible with being able to entrain to the 24-hour day. Plenty of folks walking around on this world who have periods of 24.5 hours, and they have no problem in training to the 24-hour day. And that disparity shouldn't surprise us. I showed you this individual before. His sleep is drifting about 3 hours later every day. He's got a period in his sleep-wake schedule of about 27.1 hours. We did similar protocols to assess what was his endogenous or intrinsic period. It's only 24.3. And what's interesting is that if I took anyone on this planet and I put them in a room and had them live there for many weeks like he did on the left, we would all do the exact same thing, even though our intrinsic period is only 24.3 hours. So I would argue that at least in some individuals, it's not that their period is too long. That's not a viable explanation. The other thing we did with this first individual is we exposed him to bright light. We wanted to see, is there input to that biological clock? So we studied him across 2 days at the very end of the protocol. So the gray are his melatonin levels the day before we exposed him to light. The black closed circles are his melatonin levels on the day we exposed him to bright light. And the yellow on the secondary y-axis are the actual light levels he was being exposed to. And you can see he had this robust suppression, just like Al had shown many years before, in response to bright light. So the input to his biological clock is very definitely intact and looks quite similar to those blind people who were able to entrain. So it seems like a selective loss of circadian photoreception also is not a good explanation for why this is happening. What about kind of the homeostatic regulation of sleep? Well, as part of that strange month-long protocol that we did, we were able to tease apart the circadian regulation of sleep and wakefulness, which is what's shown on the top figure. And this homeostatic component. And so this is in both graphs, it's how much wakefulness they're having in their sleep episode. And in normal healthy people, you see increasing wakefulness as they pay off that sleep drive. And compared to historical controls who we studied under the exact same protocol, there was no difference in his kind of paying his rate that he paid off homeostatic sleep drive as he slept across the night at all different biological times. So there wasn't any evidence that there was what we would call homeostatic dysfunction or a problem for the buildup and payoff of sleep drive. So that seems less likely as well. After he had his in-lab, he left us though, and then resumed his sleep at about a 24.8-hour rhythm, a little bit slower. We were able to get some of these melatonin onsets both when he was on the lab. This is again what's in red, same red dot. And you can see here on the bottom left, his clock is ticking along at a particular rate of about 24.8, and his sleep is doing that too. But what the orange arrow is pointing out is that every once in a while, his sleep suddenly moves later, and then moves earlier, and then moves much later again. He'll have these sudden jumps in the timing of his sleep. You can see that really well on day 72 here as well, where his sleep suddenly jumps, in this case, about 6 hours later in one day. That would be like a 30-hour day. And our second participant was a 39-year-old woman, and she had examples of this too. What I'm just going to show first is this is about a year of actigraphy on her. It's a lot of data, but again, the dark would be when she's moving, and the light color would be when she's not moving. And you can see, especially when we blow up and look at, say, day 50 here, you can see that the timing of activity and rest is moving progressively later every day. And if you look at these melatonin onsets, again, the red dot, they were also at times drifting later every day. Not always, but certainly when she had her most obvious periods of non-24-hour rest and activity and sleep and wakefulness. And she also had times when her sleep-wake timing drifted much faster. So what we're looking at here is a double plot, so it's the same data plotted twice, of her sleep diary data. So again, black now means sleep. The red dots are again her melatonin onset. And so you can see her drifting progressively later every day. But you can see there's a week here where her sleep-wake timing suddenly drifts very fast, where she's drifting much, much faster. And she's on much closer to a 30-hour period. Actually, it's about 32 hours periodicity. So she's on average moving about 8 hours later every day. And you can see, based on what her body clock is doing in red, she's broken apart completely from her 24-hour body clock. Her sleep-wake timing has become completely unmoored from the timing of her clock. She just spontaneously did that. And we've seen this before. So that's a clue that that might be happening in other non-24-hour folks as well. I'm going to come back to that point. She also has times when her sleep-wake scheduling is more irregular, to some extent on a more of a 24-hour schedule, although kind of delayed at a later time. You can see she's often probably averaging bedtimes after 6 in the morning. And then she'll have, again, times again, where you can see where her sleep-wake time will drift very quickly. Shifts of 6 or more hours later every day in the timing of her sleep. This is the last individual we studied in that paper. And he wasn't even a non-24 study. He was found by accident. So this was a observational study of college students that the folks in Boston were doing. This is a 20-year-old college student. And when they looked at his diaries, they noticed he was drifting later every day. He didn't have any complaints about his sleep. Again, this is sleep diary data. All of his questionnaires indicated he was actually quite happy with his sleep. But he spontaneously adopted a non-24-hour sleep-wake schedule without any complaints. And you can see, again, he sometimes had times where sleep-wake timing drifted more than 6 hours later in a single day. So what I'm going to argue is that based on what we saw, we do think that the self-selected patterns of light exposure might be shifting the clock later. And I'll unpack both of these a little bit more. So this first idea, this idea that you could disassociate sleep-wake timing from the timing of the circadian system. Well, we see that all the time in night shift workers, people who are traveling to a different time zone. We routinely will choose to sleep and try to sleep at least and be awake in opposition to our biological clock. Night shift workers do that all the time. And we do it when we travel to a new time zone. And we see this in healthy people who live in that time-free environment. So I showed this earlier, and you can see if we blow this up that there are times where sleep-wake timing drifts very fast and breaks apart from the timing of the clock, which is measured by this stippling. Again, that's when the core body temperature is below average. And you can see that sleep-wake timing sometimes is following the clock, it's following the light gray areas, and sometimes it completely breaks apart and drifts at rates of more than 6 hours later every day. And we can blow that up, and you can see here, especially around day 50, where sleep suddenly starts to break apart and drift much later. And again, these are healthy individuals who spontaneously do this when they're allowed to self-select their own schedule, their own light-dark schedule. Most of us will spontaneously do this given the opportunity. What about this idea that our light exposure pattern could be doing this? Well, this also happens normally in healthy people. And so this is where we get to Andrew McHill's studies. So healthy people on the weekends, or work-free days, our sleep-wake timing drifts later. We'll stay up later and we'll sleep in, many of us. And when we do that, we drag the timing of our clock with us because we're shifting not just our sleep, but our lights out and lights on. If I stay up late in the evening, I'm going to give myself more evening light exposure, even if it's dim light, and that will shift my clock later. By sleeping in, I'm shielding myself on, say, Sunday morning from any morning light exposure, so I don't get as much light exposure that would shift my body clock to an earlier time either. And the net result is my body clock moves later on the weekends. At least it does under electrical light conditions. So this is Dr. McHill's study. And look up at the top. So this is just a weekend. And what they did is they measured a series of participants when they were sleeping. They measured what time it was in their body, both in the weekday, before the weekend, and then after the weekend. And you can see, not surprisingly, sleep-wake timing moved later. And the onset and midpoint and offset of melatonin, you can also see them, not all statistically significantly, but certainly the onset moved later across the weekend. That's not necessarily novel. But what was incredibly novel is that then they took them all camping for the weekend. And it didn't happen. The shift to a later time is really a product of that very dim light exposure we get in the evening. If anything, they shifted earlier when they weren't getting that evening artificial light exposure while they were camping over the weekend. And their sleep didn't shift later either. So what's causing our clocks to shift later, our sleep to shift later, is partially at least our evening light exposure. And under natural light exposure conditions, we don't see that weekend shift. Now, in figure A, Monday comes along, and so we have to go to work, or we have some other commitment that forces us to get up, get that morning light exposure, forces us maybe to go to bed earlier and not get that as much evening light exposure. And so we just don't keep drifting. But this roughly hour drift per day would be non-24-hour sleep-wake rhythm disorder if it was not interrupted by Monday morning, and if I simply chose to keep on doing the same thing. And there may be some other things that accelerate this shift. So sometimes when people in time-free environments are on a non-24-hour sleep schedule, they will initiate sleep not at the beginning, near the beginning of the biological night. They'll strangely start to initiate sleep near the end of the biological night, where our body clock is somewhat paradoxically making us feel most sleepy. And when we do that, we expose ourselves to that part of our 24-hour cycle where light causes the biggest shifts in our clock to a later time. And we shield in darkness that part of our 24-hour cycle where light would shift us to an earlier time. And so we can get even larger shifts in our clock to a later time, just on where we happen to be placing our sleep relative to our 24-hour body clock. So this weekend shift that we see in response to kind of evening artificial light can be ramped up by getting even more phase delaying light in the evening, more stronger light exposure, not stronger light exposure, but light at a time in our 24-hour cycle where it's more likely to cause bigger shifts in our clock to a later time. And that brings me to diabetes. And this is where we'll end. So diabetes is two disorders that we give a single name. So there's type 1 diabetes. What's that? Well, this is an autoimmune disorder. So you have T-cell mediated destruction of the beta cells in your pancreas. Without those beta cells, you'll have insulin deficiency, and you'll become hyperglycemic because you no longer are making insulin to decrease your blood sugar levels. This is a loss of function disorder. This is like what happens to non-24 and blind individuals. Instead of loss of pancreatic beta cells, we have loss of photic input to the circadian pacemaker. That results, in this case, in an absence of circadian resetting by light and a loss of entrainment. These are both loss of function disorders. But we call something very different diabetes as well. Type 2 diabetes, which is less and less adult onset diabetes, is an environmental and behavioral disorder. We live in an environment with an overabundance of food, which is in stark contrast to the food scarce environment that humans evolved in. Plus, we have low levels of physical activity. That's a behavioral change. That leads to a biological change, excessive accumulation of body fat. That in turn leads to yet another biological change, insulin resistance. And in some individuals, now we actually end up with frank insulin secretory failure. But the underlying etiology is an environmental and behavioral one. We call these two disorders diabetes, but they're really very different disorders that kind of look a little bit the same in the end. It's a behaviorally and environmentally induced disorder. Non-24 in the cited, it looks like this is an erroneous slide, is also, in our opinion, in some individuals, a behaviorally and environmentally induced disorder. In the cited individuals, it's an environment with not an overabundance of food, but an overabundance of evening light exposure, which is in stark contrast to the evening light scarce environment in which humans evolved. The sun went down and you had a tiny bit of campfire light. But if you look at studies of hunter-gatherers, that's actually quite dim. Plus, we have a behavioral change of low levels of morning light. And that leads to circadian phase delays, a biological change, which leads to where the X should be a loss of entrainment, another biological change. And so what we have proposed is a change to the medical classification or the nosology of non-24 that would be very analogous to what we have for diabetes. We have two types of diabetes, type 1 and type 2. And we really should have two types of non-24. That in the blind, which I would call type 1, and that in the sighted, which I would call type 2, that have very different etiologies and may sometimes look similarly. And with that, I would just like to thank all the folks who have been working on this for a very long time, both in Boston and here in Portland, as well as Baltimore. So thanks very much.
Thank you very much, John. That was great. There are a couple of questions in the chat. I'm going to start with the one that I tried to type. It's a long one, but it sort of follows on from your analogy with type 2 diabetes. The question I have is, apart from in somebody who has non-24-hour or who is blind and therefore is not synchronized with the environment in terms of their rest activity cycle, fasting feeding cycle, for very long periods of time, and that's in a variable way. Does that have other consequences, do you know? Of course, it affects sleep continuity, like you mentioned. That probably has effects on cognition and mood, you could imagine. But there could also be adverse health effects in this population. Do you know anything about that?
Yeah, so it's a great question because, of course, you have shown that if you take a sighted person and you kind of misalign the timing of their clock to the timing of everything else, food intake, sleep, activity, you do have adverse cardiometabolic consequences. And so the data on this is not really good. There's not been a really good systematic survey of blind individuals with non-24 and those blind individuals without non-24 and comparing cardiometabolic outcomes.
Mm-hmm.
So the short answer is I don't know. I would say empirically, I think there was a lot of obesity that I saw, both in Boston and here in Portland. And I would not be surprised if you compare it to blind and totally blind individuals without non-24, that there were in particular metabolic consequences. A lot of them would get placed on thyroid replacement as well. That I think actually is a confounder, however. I don't think there's any direct thyroid implication, but I do suspect that when I'm out of sync and I'm 188 degrees out of sync with the 24-hour day and I'm feeling tired during the day, if I'm a blind person with non-24, I go to see my doctor, say I'm really tired. Oh, well, maybe you're anemic or maybe you have low thyroid. Let's check your thyroid, your TSH level. Well, it turns out TSH has this, strong circadian drive to peak in the biological night. But if I'm out of sync, now I've got this peak for my TSH to peak in the middle of the day, right when my doctor is measuring it. They say, oh, your TSH is high. That must mean you're hypothyroid. That's why you're so tired. And so I do wonder if in addition to kind of cardiometabolic problems, there might be iatrogenic, position-induced problems, because we don't realize that they're actually out of sync. So I would say your question's a great one, and something that really, really needs to be studied. Because I would suspect that the kind of cardiometabolic consequences could be even more significant than the impact on sleep.
Thanks, John. Stephen Lloyd has a question. Don't know if you want to ask it in person, Stephen, or I can read it out.
It's probably a bit off topic, but say for individuals who may be working on, say, an orbiting space station in which they are zipping around the world, multiple times a day. Have people look? I mean, I'm just wondering whether their melatonin secretion levels would be also drifting similar to that of someone as a blind person, because they're probably on a 90-minute day. I'm not sure how many times they're
Yes.
but so I'm just wondering, I would imagine for short-term space travel, this is probably not that big a deal. It's more like kind of a jet lag. But for those people who are spending, 6 to 18 months on an orbiting space station, how is the drift in the circadian being affected in those individuals?
So that has been studied, and it depends. So if I was exposed to that 90-minute day, then yes, I couldn't synchronize to it, and I would drift just like a blind person. But they're not, of course, they're controlling their lights out and lights on, right? And they're living on a 24-hour schedule of lights out and lights on within the space station. And when it's their nighttime, their eyes are closed and they're in a dark environment. And when it's their day, they're exposed to primarily artificial light with whatever has kind of come through the portal or the cupola on the space station or something like that. That's one piece. However, if you look at submariners, so a guy named David Neary did a study many years ago looking at people who were living on submarines who had a duty schedule of, I want to say, an 8-hour day. And they did exactly what the blind people did. So similar to a 90-minute day on a space station, if that's what I was exposed to, they were unable to synchronize. And this was a nuclear-powered submarine with nuclear missiles on it. And none of these people, none of them, I think, well, certainly none of them were entrained to the 8-hour day, and none of them were entrained to the 24-hour day. They were all what we used to call free running. They were none of them were entrained. And he physiologically measured the time of their clock. So yeah, so that's what will happen, whether you're in space, if you're getting a true 90-minute day, or if you're on a sub.
John, can you just clarify that on those nuclear subs that you took about an 8-hour day? Because I thought that they were on 18-hour days.
Yeah, I forget actually in David's study what it was. Since I've come to work at 10 years ago at the VA, I've had veterans tell me, oh yeah, well, there's the 18-hour day and then there's the 12-hour day. So I think it maybe have been an 18-hour day in David's study, but there are a lot of these very different in the military, these kind of very odd duty schedules, particularly in the Navy. So you may be right. It may have been an 18-hour day.
Well, just for further clarification, an 8-hour day, some people think of an 8-hour work day, but what you would be referring to, or even an 18-hour day, would be an 18-hour
4 hours on, 4 hours off.
Sleep-wake cycle, which incorporates sleep, which would be, if it's an 18-hour day, it would be 6 hours of sleep and 12 hours of wake.
Exactly.
Yeah.
So so apparently there's some very odd duty schedules out there.
Derek Lamb has a question. Derek, do you want to ask it? That was the question that we had before the talk started. So let's have an answer.
Sorry, I tuned in late, so sorry if I missed the discussion.
No. This was before it started. So
yeah, I was just wondering what your thoughts were about daylight savings and whether it matters. Maybe an hour difference doesn't make that much difference, but and then
it does. It makes a huge difference. So there are two answers to that question. One, the idea of shifting across the year is a bad idea. So one of my favorite studies is a New England Journal of Medicine study where they looked at about 10 years of Swedish Health Registry data. And they looked at the 2 weeks prior and 2 weeks after the spring daylight savings time shift. And there was an increase in myocardial infarction on the Monday, Tuesday, and Wednesday after the spring shift. So that would be an argument against shifting. However, that all said, I would recommend shift it. If you're going to stay on one time, you would want to stay on standard time, not daylight savings time. And here's why. If you go to daylight savings time, you're increasing evening light exposure and decreasing morning light exposure. That will help push your clock to a later time, which is likely going to be worse from a psychiatric and kind of perspective, at the very least. So I think what you would want, we already have this propensity to phase delay anyway, shift our clocks to a later time, which is what we've been talking about all today. I think the last thing we want to do is encourage that. So I would say no shifting, and if you're going to pick one, I would pick standard time. And there are a lot of other health outcomes that you can show get worse as you kind of delay. And there's like time zone, within time zone data that's very interesting along those lines.
And so it seems like that's what I have read, for most people seem to advocate for keeping standard time. And as I understand it, the bill that is being considered is to keep daylight savings time. I'm wondering why that is.
Yeah, that's a great question. And I will not claim to be an expert on the matters of congressional debate. So
A fascinating talk. Thank you, John.
thanks.
Yeah, thanks, John. So it's gone 1 o'clock. We had 51 participants, which is a great turnout early on. I think there's about 33 now. So people are probably going to jump off, but I do want to ask for one more round of questions, if there are any questions. If you've got a few minutes, John.
Yes, happy to.
And if there are any questions, chime in now. And if there aren't any, I just want to thank you so much, John, for a great talk. Really very stimulating. And I think Doris has got a question. Maybe?
Yeah, I was kind of about that daylight savings thing. Does anyone know if there was any input from people who are involved in sleep studies and these kind of things to the Oregon government? Did they even ask anyone about the consequences for that? Does anyone know?
I don't. No one asked me.
So there's a government advocacy group attached to the Sleep Research Society and the American Academy of Sleep Medicine. Both of them have contacted, I think through NIH, but maybe through government, straight to the government as well, advocating for continuous standard time. And it was totally ignored, of course. But the fight is only just beginning. I think this one was snuck through. It was a very unusual thing with bipartisan support, and it wasn't on the agenda, and someone threw it in at the last minute, and nobody knew. Everyone had slept in that day, and all the people who would normally oppose it were still asleep, and so it went through. But that was it the Senate, but and I mean the House, but now it has to go to the Senate maybe. So we will see.
So hopefully we'll stay on standard if we have. The thought about getting up in winter, when it's pitch dark, is really not very appealing.
Yeah, having the kids get up that early is tough. It used to be tough, but now it's going to be tougher. I feel. Thank God my kids are grown up and have to get themselves up now. All right. Well, John, thanks very much. It was greatly appreciated.
Great. Thanks.